No, I'm good. Yeah, thanks.
Oh.
Should I switch out?
No.
What is this?
This is for the house.
Oh, true.
If you need it.
Yeah. Does that one also work?
That would also be for the house. That would be in case that one dies.
Oh, cool. Yeah, so we see it's not really working. Aha. Yeah, so, oh my goodness. It's pretty loud. Yeah, so sister chromatids are seen after S phase, and that's when we get DNA. Do I have to use this? Okay, it's kind of loud. We see that after S phase in the cell cycle, and that's when you see the chromosome go from this guy to these guys, right? This is S phase. And remember, they're both considered one unit, one chromosome, except this guy has one DNA molecule, this guy has two DNA molecules, right? And then it wouldn't be parent cells in the fertilized offspring because, yeah, the fertilized offspring is obviously the combination of two parents, and then one parent would only be like half, right? So now we'll ask ourselves, what does it mean to be diploid or haploid? It's a pretty common definition, but I feel like people still have trouble defining it. Would someone like to take a go at it? Yeah, like so your diploid number, right? Different organisms have different diploid numbers. What could I represent the diploid number as? 2n, really good, thank you. And then n would be haploid, right? So if you want to find an organism's haploid number from a diploid number, just some really simple algebra, you obviously just divide by 2. And a diploid organism has two sets of genetic information, right? So in diagrams, you probably see like a blue chromosome and a red chromosome, one representing maternal, one resembling paternal. And the diploid organism will have homologous chromosomes, right? And the haploid individual only has one copy of all the genetic information. So we'll look at number 3 now. We're looking at the cell cycle. Where do I have growth? In which phases? Could someone tell me what G0 means?
Indefinite side phase that just cycles continuously without meiosis.
Yeah, really good. Thank you for that. So G0 is when the cell leaves the cell cycle. It's not really growing or dividing anymore. G0 is present in like brain cells because your brain cells really aren't dividing anymore. But we have growth in G1 and G2, right? Because we could think G stands for growth. And as it was said earlier, S phase is the phase of DNA replication, right? And then M phase is obviously, we could think of it as mitosis or meiosis. This is the actual division of the cell. And everything that's not M phase, we could represent as interphase, right? An interphase is just when the cell is living its life, growing, replicating its DNA. preparing for division, and it spends most of its life in interphase.
So you would say growth is happening in S phase as well? Or not?
No, growth is not. Growth is happening in G1 and G2. Because G1, the cell is preparing to grow to replicate its DNA, right? And then G2, it's preparing to divide.
So replication isn't considered growth. Okay.
You bet. What the? And for you guys that came in late, if you want to pull this up, it's on ucfstudyunion.wordpress.com, or you can just follow along. So number 4 is one of those chromosome number problems, if you guys haven't forgot how to do that already. So our cell is in the G2 phase and it has 16 chromosomes. In metaphase of mitosis, how many DNA molecules does this cell have? 32, right off the bat, thank you. So yeah, we know there's 16 chromosomes and we know the cell is in G2, which means that the cell is past S phase. In S phase up here, we get 2 DNA molecules per chromosome. So this cell would have 16. Now, yeah, if this cell was in anaphase of mitosis and it just finished, how many chromosomes would I have? (32)? Yeah, you would, right? Because in anaphase, that's when the sister chromatids separate. So you now get, this guy actually goes back and then turning into 2 of these guys. And they're still in the same cell because it's just after anaphase. We don't have cytokinesis or telekinesis that's happened yet. So our chromosome number would double and our DNA molecules would stay the same. Do you have a question?
At what stage, like after G2, does it turn into?
Yeah, that's a really good question. So G2 going into M, the chromosome number is pretty much going to stay the same until anaphase, right? Because anaphase is the first event that really has the sister chromatids separating. If we're thinking about mitosis, prophase, nothing's really happening. Metaphase, they're just lining up on the plate, but then it's anaphase where they actually separate. So that's when you're going to see a change in chromosome number. For this question, it's 32. Do you see how that is?
Oh, because there's 2 DNA molecules per chromosome?
Exactly, because we're past S phase now. Yeah, so that kind of leads into the next question. When do chromatids become chromosomes, and what effect does this have on the number of chromosomes in a cell? So that's pretty much what I just talked about, and that is in anaphase, right? Because we can consider this unit one sister chromatid, or 2 sister chromatids, or 1 chromosome, right? So these chromatids, the sister chromatids, become their own chromosomes after anaphase, because that's when. then we can consider one chromosome has its own centromere, right? One centromere to a chromosome. So yeah, number 6, there is a nice formula for this question. Does anyone remember what that is?
To the n where n is the homologous pairs.
Exactly. Thank you for that. So just to repeat that, how can we calculate the possible number of chromosome combinations? It's 2 to the n power, where n equals the number of homologous pairs. So number 7 is asking, what is considered to be the fundamental difference between males and females of most organisms? Gamete size, yes, really good. So yeah, males produce relatively smaller gametes, while females produce larger ones, like sperm and egg, right? So how does anaphase 1 of meiosis differ from anaphase of mitosis?
In meiosis, homologous pairs separate. In mitosis, sister chromatids separate.
Yeah, really good. Thank you for that. So yes, in anaphase 1 of meiosis, the homologous pairs will separate because in metaphase of meiosis, remember the homologous pairs match up with each other and eventually they will segregate, right? The law of segregation. And in anaphase 1 of meiosis, since we're having the separation of homologous pairs rather than sister chromatids, after anaphase 1 of meiosis, you're not going to see a change in chromosome number, right? Because our sister chromatids are still together. But in anaphase 2, you would see a doubling of chromosome number because that's when we get the separation of sister chromatids. So who knows number 9? D? Does everyone agree with D? Cool. Because we can rule out interphase and mitosis because that's not crossing over, never occurs there, right? So what happens during crossover? Does this generate new alleles? If not, what does? So recombination, right? What does it occur between in meiosis? homologous chromosomes. Yeah, really good. It doesn't occur between sister chromatids, right? And does this generate new alleles?
No, it generates new combinations of alleles.
Yeah, really good. Thank you for that. So it's kind of shuffling the deck of cards here. We're not producing new alleles, and that is done by mutations, right? And so, yeah, crossing over is recombination of the two non-sister chromatids of a homologous pair. And once again, only 2 out of the 4 chromatids can participate in recombination. So it's kind of back to chapter 7. That's why you have a maximum recombination frequency of 50%, because if it happened every time, still only 2 out of 4 of these chromatids would participate in crossing over. Yeah, that's a really good question. So each of the homologous pairs, there's they have different versions of the genes, right? Which we call alleles, and one is located on one, and the other is located on the other. And crossing over, they'll be swapping alleles from each other, so we'll create different combinations on a single chromosome. But we're not generating new alleles, right? We're just mixing them up together. So who could define chiasmata? Is it A? It's D. Okay, so it's the area where crossing over occurs, right? It's not A. A would be what? Centromere, yeah. And then sister chromatids don't have any differences between themselves. So we're thinking about Shigoshin now. What do you guys think the answer would be? B? Okay, yeah, really good. So the answer is B. Its function, does anyone remember?
It keeps the separates from separating sister chromatids during meiosis 1.
Yeah, exactly. Thank you for that. So like what he said, the function is to keep the sister chromatids in anaphase 1 of meiosis from separating, because in anaphase 1 we're having the separation of homologous pairs. And if we had our sister chromatids separating in anaphase 1 of meiosis, we would get some aneuploidy, and that would not be good. So how many cells are created after meiosis 1 from one cell? 2, right? And are they a haploid or diploid? Haploid? Yeah, thank you for that. They are haploid because in anaphase 1, our homologous pairs separate, so then the law of segregation states that one cell will get one allele, basically that the alleles will segregate. So then after meiosis 1, we'll have 2 haploid cells, right? Because each cell only has one copy of the genetic information, right? It doesn't have a maternal and a paternal, only has one or the other. Then at the end of meiosis 2, we'll get 4 haploid cells, right? And then whether or not, if it's reductive or equational, that's just something you'd have to just know, right? So meiosis 1 is reductive, and then meiosis 2 is equational. So the answer would be B. So what would 14 be? It is A. Nice job. So it is a form of a gene found at a given locus. And we could define C, the location of a gene along a chromosome, as locus, right, or loci. So if your genotype is YO, how many sex chromosomes do you have? You have one, right? Because the O is representing that something happens in meiosis to where you didn't get your second sex chromosome. So which one of these would that be indicative of? Excuse me?
Lethality. Because you can't survive without an X chromosome.
Yeah, exactly. So it would be lethality. Your X chromosome has the majority of the genes you need to survive. Well, the Y chromosome is more there to indicate which sex you're going to be. And then remember, in the Y chromosome, it's a lot smaller. And we have that thing called SRY, sex determining region Y. And that is the thing that will dictate one's gender biologically. Klinefelters, does anyone remember the sequence of what Klinefelters would be? XXY, yeah, really good. Then triple X, it's quite literally triple X, so it'd be 3 X's. And then Turner syndrome would be XO, right? And then the super male syndrome would be XYY. Yeah, really good. Thank you for that. So we see the XXXO system in those types of organisms, right? Insects, grasshoppers, crickets. And in this case, it's a bit different in that sex. So when we're thinking which one is the homogametic sex, we're thinking, all right, which one is the one that's producing one type of gamete? And in this case, the females are actually the XO and the males are the XX, right? In this system, it's flipped. So yeah, the XX individual, in this case it would be the male, is the homogametic sex, meaning that he only produces one type of gametes, right? So this individual is only going to produce gametes with an X in it. The XO individual would be the heterogametic sex because half of her gametes would have an X and half would have nothing, right? The O.
What's the other sex determinant system, the ZW?
Yeah, that would be the ZZ and the ZW.
Is ZZ female or ZW?
Female ZW. Okay. So then the male in that case would be homogametic and the female would be heterogametic, right? So who thinks they know the answer to this one? C is a good answer, but it's actually D in this case. Because C is saying that the sexual phenotype is determined by the combination of sex chromosomes present, but in the case of genic sex determination, it's determined by genes, right? Not actually what their chromosomes are. So try to work this one out by yourself, and then we'll compare answers in about a minute or two. Does someone want to say what they got?
Divided by 32.
1 over 32? Okay, did everyone else get 1 over 32? So if I was going to solve this, there's a few different ways to solve this. If you want, this is the and multiplication rule, right? So I'm saying what's the probability? I would get that genotype for A, B, C, D, and E and F in my offspring, right? So I would calculate each individual probability and then multiply them all because they're independent events. So if I was finding the probability that the offspring would get homozygous dominant A's, and I'm looking at the parents. So the first parent is heterozygous. I need 2 big A's. So he has a 1/2 chance of giving his big A, right? And the other parent has 2 big A's, so he has a 100% chance to give that A. So that's going to be at 1/2 chance. And I look at the B's. What's the probability I'll get 2 big B's? And the first parent only has big B's, so that's 100%. And the second parent has 1 big B and 1 small b, so that's 1/2 a chance he'll give a big B, right? And then I look at the C's. Same thing for the A's and B's. We need 2 big C's. The first parent only has big C's, so that's 100% chance. Second parent only has 1 big C, so it'd be a 50% chance. And then for the D's, we need a heterozygous individual. And one parent only has small d's, and the other parent only has big D's. So that's going to be a 100% chance. And then for the E's, same thing. And then for the F's, it's the same thing. So is everyone good with this one? If you got like an impossible one, like they would ask for like a homozygous recessive type for a trait, and none of the, like there's not, what am I trying to say here? They're asking for a homozygous recessive trait, and you don't have 2 little alleles possible, then the whole thing would equal 0, right? So what do you guys think the answer would be here? A. Okay. Does everyone agree with A? So in a group of 30 turtles, 10 have no green pigment, 12 have a lot, 5 have a moderate amount, and 3 have very little, right? So we see some expressing it, and we see some not expressing it, and we see some expressing it more than others, right? So variable expressivity, that would be correct, because different turtles are expressing the trait to a different degree, right? So that's what we can think about that one. But it's also variable penetrance as well, because we see penetrance are the people with the traits expressing it or not, right? It's not to what degree are they expressing it, but it's whether or not they're expressing what they should, right? And in this case, 10 turtles have no green pigment, but others do. So they probably have the genes, but they're not expressing what they should, so it's not penetrating, so there is indeed variable penetrance, right? Then it would not be incomplete dominance, because that's, remember, it's intermediate, right? Heterozygotes are having an intermediate phenotype, and then codominance, they would be codominating, so the heterozygous individual would display traits from both alleles, and it wouldn't be mixed. So this one would be both A and B. Yeah, you can't really see, the formatting's kind of bad on this one, but which appear approximately equal in males and females. it would probably just, which ones wouldn't it not be? X-linked recessive, nice. So it wouldn't be any of the sex-related ones, right? So any X-linked or holandric, which is another term for Y inheritance. Obviously, since they're linked to sex, we're not going to see them equally in sexes, right? So it would be autosomal recessive and dominant. So yeah, which of these forms of inheritance skip generations? In her Pedigree PowerPoint, remember she has that page of each type of inheritance and certain inheritance patterns you can expect to see. In a Pedigree, it'd probably be of your best interest to know that, because that's a good way she could ask questions regarding it, right? So each one of these would have like 3 or 4 listed bullets, and she could just ask a question, which one of these is this bullet? You should know the answer to it. So in this case, which forms of inheritance skip generations? Which ones do you guys think? The recessive ones, right? So autosomal recessive and X-linked recessive. So all of the dominant and holandric traits we'd expect to see in every generation, right? But in the case of autosomal recessive, if I was looking at a pedigree and I saw that this trait did skip generations, I could rule out that it's a type of dominant inheritance, right? So the next thing I would look at if I'm looking at a pedigree would be, do I see it equally in both sexes, right? If I see an equal number of males and females affected, I would expect it to be autosomal recessive. And if I saw a different proportion of males and females affected, I would suspect it would be sex-linked, right? So in the case of complete linkage with no crossing over, the progeny of a heterozygote and homozygote are Yeah, really good. Thank you for that. So it is E. To answer this question, you would need to know what complete linkage means, right? So completely linked. We're thinking about the word linkage, right? Linkage is meaning what? These genes are what to each other if they're linked genes? Close proximity, okay. Thank you for that. So if we're saying they're close together, we're probably thinking that they're on the same chromosome, right? And if they're on the same chromosome, in the case of complete linkage, they're so close together on the chromosome that crossing over is statistically not going to happen because they're so close, and we know that crossing over occurs at a random part along the chromosome. So in complete linkage, they're so tight together that we're never going to get a crossover event occur between these two genes of interest, which means we're not going to get any recombinant progeny if we're not having crossing over, right? So we would not see any recombinant offspring. We would just see all non-recombinant with the parental phenotype of 1-to-1, right? And in the case of unlinked genes or independently assorting genes, we would see a gene ratio of 1-to-1-to-1-to-1, right? Because if we're acting like they're completely unlinked or independently assorting, we will see an even amount of recombinant to non-recombinant offspring, right? And if they were linked but not completely linked, we would see a greater number of non-recombinants and a lesser number of recombinants. So I remember on one of your exams, she had that question where she had the picture of the structure of the nucleotide, and she asked you to identify if it was ribose or deoxyribose and a pyrimidine or a purine, right? So if I'm thinking, what are the components of a nucleotide monomer? What would they be?
Sugar, phosphate, and nitrogenous base.
Yeah, really good. So we'll have our sugar, and that will be in the middle, and attached to that will be a phosphate and the nitrogenous base. And then how we differentiate between deoxyribose and ribose. So, excuse me? Hydroxy group, yeah, really good. At which position? Third carbon. Almost. It's the second, right? Thank you for that. So yeah, ribose and deoxyribose, we could think deoxyribose is ribose but deoxygenated, right? All nucleotide monomers will have a free 3' hydroxyl group, and that's what is key in the addition of the polymerization of nucleotides, right? Nucleotide monomers are added to free 3' hydroxyl groups. But in this case, what differentiates the two sugars is the presence of a hydroxyl group at the position 2', right? And the difference between a purine and a pyrimidine, one has 2 rings and one has 1 ring, right? I like to think of it, purine has the shorter name, but it has the more rings, and the reverse for pyrimidine, right? Pyrimidine, longer name, but 1 ring. Yeah, and purines are also adenine and guanine, and then pyrimidines would be the remainders, right? So we just talked about what a nucleotide is, right? It's a nitrogenous base with a ribose sugar and a phosphate group. But now we're differentiating between a nucleotide and a nucleoside. So a nucleoside has something extra, or it's missing something? It's missing what? Nitrogenous base. Okay. Does everyone agree with that? Phosphate? So we have two different answers here. What do you guys think? Phosphate? So it is phosphate, right? A nucleoside is just a base and a sugar. It is lacking a phosphate. And you, sometimes it's said a nucleoside triphosphate. It's pretty much a nucleotide, but you're kind of segregating phosphate in the actual nucleotide.
So the nucleoside's got more phosphates?
It doesn't necessarily have more phosphates because technically a nucleoside is without phosphates, but some people like to say nucleoside triphosphate, and you're thinking what's the difference between that and a nucleotide, because a nucleotide is including phosphates, but it's just a different way to name them. So who thinks they know the answer to this one? It's A. Does everyone agree with A? So it is A, right? So it's only new strands on the leading strand are synthesized in the same direction as the movement of the replication fork. That is what we would define the leading strand as. It is Synthesis is in the direction of the replication fork, while the lagging strand is in the opposite direction. So that's why B would be incorrect. And then in eukaryotes, there are multiple origins of replication. So C would not be correct. And synthesis is only in the 5 to 3' direction. It's the same for transcription as well. You're only seeing polymerization in the 5 to 3' direction. So the answer would be A. So the function of RNA depends on what? So it would be its secondary structure, right? So the things we talked about, like stems, hairpins, and loops, these are all going to affect what function the RNA has in the cell. And the central dogma of biology, does anyone remember what that is? Yeah, really good. So it's DNA to RNA to proteins, then DNA is obviously replicated by DNA replication, then DNA to RNA we would call transcription, and RNA to proteins is called translation. So it's meaning that all of our genetic information that we have in our cell is in our DNA, and in the DNA we have genes, and genes code for functional products like enzymes that our body uses. So it's just saying that we get proteins from our DNA, and it's a pretty sophisticated system. Also, sorry if you're following along, the numbering is kind of messed up because I hit enter a bunch of times. So which of these enzymes involved in DNA replication has proofreading capabilities? Okay, polymerase 3 and Yeah, so do you guys agree with polymerase 3?
That's what polymerase 1 gets rid of the primer.
Gets rid of the primer? Okay.
Polymerase 3 lays down the RNA or the DNA.
Yeah, so in this case, actually, polymerase 1 and 3 both have proofreading capabilities, right? In different directions. So the function of DNA polymerase 1, does anyone remember? Nobody? The function of DNA polymerase 1 binds to the initiator. Does not. But thank you, though. Okay, and it replaces it with nucleotides, right? Okay, really good. Thank you for that. So yeah, DNA polymerase 1 does remove the RNA primers and replaces it with DNA nucleotides. What you were thinking of, I think, for the initiation of DNA replication, that's when the initiator protein binds to the initiator region and kind of causes that short stretch to unwind. And then from there, you have your single-stranded binding proteins and helicase come in. But the function of DNA polymerase 3, would anyone like to share?
It's to lay down the new strand of DNA.
Yeah, thank you for that. Really good. So it's doing the main polymerization and DNA replication. That's what's laying down the new nucleotides. Then DNA ligase seals the nicks, right? Because after DNA polymerase 1 comes in and replaces the primers with DNA nucleotides, you still have gaps in between where the primers used to be. So DNA ligase is going to connect them together. Then primase is our primer laying enzyme, and it's going to lay down RNA primers to initiate the polymerization of our new strands. And this is required because what? Why do we need primase? Because our DNA nucleotides need a free 3' hydroxyl to be attached to, right? And the RNA primers from primase allow that to happen. But then it's RNA, so that's why we need DNA polymerase 1 to come in and replace our RNA with DNA, because we don't want RNA in our DNA. And then gyrase is also known as topoisomerase, and that could be used to relieve supertension or supercoiling ahead of the replication fork. Yeah, really good. It is C, right? So which statement best describes the transcription apparatus most commonly seen in bacteria? That would indeed be 5 polypeptides and a sigma factor to form the functional hollow enzyme. So which statement is incorrect regarding row-dependent termination? B? Yeah, really good. So it is B. Where do we see a rut site? Upstream. And do we see it in row-dependent termination? No, right? It's in what? Row independent. So the function of the RAT1 protein. C? Yeah, nice job, guys. So it does operate in eukaryotic termination. So I think it's important when you're going through and reviewing, especially for like the exam 4 block, there's a lot of similarities and differences between eukaryotic and prokaryotic methods, such as like termination and whatnot. So it's kind of helpful to know what they have in common and what they differ by. So where can I find a 5' cap? mRNA? Yeah, so we find it in mRNA. Thank you. And what exactly is the 5' cap? It's the type of nucleotide, right? And we could say it's a modified guanine nucleotide added to the 5' end. And we find it in mRNA, so we're not going to see it increasing tRNA stability. It would increase mRNA stability. And yes, so the answer would be what in this case? C. Yeah, nice. So it functions to stabilize mRNA, but also allows the recognition of mRNA by the small ribosomal subunit. Yeah, and our other modification to our mRNA would be the poly-A tail, right? And the removal of the non-coding regions. And note that transcription and translation in eukaryotes they happen in different locations, right? So transcription is involving DNA, and DNA is located in the nucleus. So transcription happens in the nucleus of the cell. Then our ribosomes are located in the cytoplasm. So that's why our mRNA has to be processed, because it's leaving the nucleus and entering the cytoplasm. So we want to make sure the message is stable. But in the case of prokaryotes, transcription and translation is coupled. So they're happening simultaneously. And prokaryotes don't have a nuclear envelope or organelles, so they're kind of, it's happening at the same time. And that's why these modifications are not made in prokaryotes. So yeah, there's different types of RNA polymerases that take part in transcription. You just have to know which one. It's just a matter of 1, 2, or 3. So DNA polymerase probably isn't going to take place here, because we're talking about translation and not replication. And in transcription, sorry, I was talking about transcription, not translation. And in transcription, we're making RNA, so it's probably not going to be anything with DNA in it. So now, is it 1, 2, or 3? That's the question. 2, right? Yeah, it is 2. Really good. RNA polymerase 2, so B. So now, why do we consider the genetic code to be redundant? Yeah, really good. Thank you for that. So the answer is E. Amino acids may be represented by multiple codons. And note that one codon does not code for more than one amino acid, right? One codon only codes for one amino acid. So in eukaryotes, where can I find the start codon? A, yeah, really good. So the Kozak sequence. Once again, this is one of the things for exam 4 where you kind of just have to look out for, okay, is this in eukaryotes or prokaryotes? Or do they have that similar or different, right? So it's actually misspelled in here, but the Shine-Dalgarno sequence, what is that to, what is that?
It's in prokaryotes.
It's right, it's the start codon in prokaryotes, right? So, yeah. So IF3 has what function in translation? Yeah, really good. It is C. So IF3 does prevent the binding of the large and small ribosomal subunits during initiation. Yeah, so an operon related to an important enzyme has a regulator gene that codes an inactive repressor. What type of operon is this? And I know she didn't really include positives in your guys' class, so we don't really have to worry about those. All we have to look at is whether or not it's repressible or inducible, right? So it's coding an inactive repressor. So what do you guys think? Is it repressible or inducible? Inducible? Does everyone agree with inducible? Repressible? Okay, why do you say that?
It's encoding for a repressor, meaning like it's able to repress, meaning that it's in the opposition.
Okay, yeah, really good. Thank you for that. So we see that since it's coding for a repressor, it would be negative, right? So now the repressible or the inducible part, it just has to do, is it off or on, and are we turning it off or on, right? So in this case, it's inactive, so the gene, it would probably be on, right? If there's an inactive repressor, it's not repressing, it's on. And since we can activate the repressor with the co-repressor, we can activate repression, so it is repressible. Right. So I think repressible, we're able to repress it. And if we're able to repress it, we're able to turn it off. So it should be on. And if it was a negative inducible, the repressor would be active. Right, because we can induce this thing to be on, so if we can induce it to be on, that means it's off. And if it's off, it probably has an active repressor initially. For this one, it is negative repressible. This is actually, yeah, we'll skip this one. So we're going to skip 38 because that one is involving a positive one. So which cis-acting sequences in eukaryotes are position and orientation dependent? Is it promoters? It's actually not promoters. But thank you for that. It is enhancers? Yeah, really good. Is that all? Are silencers position and orientation dependent as well? I see some agreement. So yeah, they are, right? So the answer would be both A and C. So now we're asking ourselves, what modifications can be made to histone proteins? It can add something to it, or maybe remove something from it. So this is oh, sorry. No, you're good.
Reacylated or deacylated causes transcription to decrease with
Yeah, awesome, really good. Thank you for that. So to histones, we can add acetyl groups, and this will affect the rate of our transcription. We know that histones have positively charged tails. We know that DNA is negatively charged. Yeah.
Sorry, what was the answer for the previous one?
It was D, both A and C.
Well, when you have your slides, it's written that enhancers are orientation independent.
So really? Then it would just be silencers then. Yeah, thank you for bringing that to attention. So it's just been found that enhancers are orientation independent, so scratch what I said. Sorry about that. In this case, yes.
But promoters are orientation dependent.
Dependent. Are they both position dependent though?
Yeah, it's written over here, orientation position dependent promoters.
Cool. Thank you for bringing that up. So apparently I was wrong again, and that promoters are indeed position orientation dependent.
So it's B and C.
It would be B and C in this case. Yeah, so histones, back to that, thank you for that. They can be acylated or deacylated. Histones are positively charged. DNA is negatively charged because of its phosphate, its free phosphate. So they attract to each other. And when you acetylate the histone itself, you're not acetylating the DNA. You're going to make the histone less positive, so there's less association between the histones and the DNA, more transcription, and then histone deacetylase will remove them, less transcription. So what attracts chromatin modifying proteins to restore chromatid modification on newly synthesized DNA? So we're thinking about chromatin now. So what types of modifications? Oh, yeah. Yeah, methylation, right? So methyl groups promote or inhibit transcription. Inhibit, right? So you can think that the methyl group is sticking up on top of a DNA base and it's preventing enzymes from coming in and doing what they need to do. So what attracts chromatin modifying proteins? That would be hemimethylated DNA, right? Because we know in DNA replication, it is semi-conservative, so each of our new strands are going to be half parental, half daughter. And so if it's half parental, then half of the new DNA will be methylated. So that's why we're saying hemimethylated DNA is attracting these modifying proteins. And the modifying protein in this case would be maintenance methyltransferase, right? So that's going to restore the methylation pattern to our new strands of DNA. So what in eukaryotic DNA allows a mismatch repair in a cell to distinguish between the old and the new strand? Okay, yeah, you're right. The old strand is methylated, but we're thinking more about in replication, like if we get a mismatched nucleotide base, right? And we need enzymes to go in and figure out which strand is the old strand and the new strand. Because we don't want to modify the old strand because that would just completely throw off our DNA, right? We want to fix the new one that we've placed. So how can the cell distinguish between the old strand and the new strand? It is methylated, you're right. But in this case, enzymes would search for nicks in the strand, right? Because you know in replication, we have nicks. Yeah. That's something else. Yeah. So what modifications are made to pre-mRNA to produce mature mRNA? It was nicks in the new strand. So does anyone want to tell me one modification? Yeah, good. The 5' cap, and then you said poly A tail, right? And then one more thing. Yeah, really good. Yeah. So introns are removed. Introns are non-coding regions. Exons will be kept. And then if you remember, we use that thing called alternative splicing, and that is going to be generating a variety of proteins from one gene, and that's just by keeping or removing certain exon regions from the gene, right? And now we'll think about epistasis. So what is a recessive epistatic gene? Yeah, exactly, really good. So epistasis is about masking. So in the case where we have a recessive epistatic gene, this gene will be the recessive, right? So if I have two, if I'm homozygous recessive, for this trait. A really good example would be the Labradors, right? So there's two different traits, right? Like B and E for hair color. And if one is the recessive epistatic gene, if it's present, it'll mask the expression of whatever the other gene's saying, right? So that is all the questions I had prepared for you guys today. I want to thank you all for coming out, because this was really early. So you guys are awesome. Thank you. And for the exam, what are you guys going to do to study for it? Because it's a lot of content, right? PowerPoints, right? I think PowerPoints are the most important thing. She does a really good job of bolding words you need to know, right? So if you're just trying to do a crash course, maybe try making Quizlets or flashcards about all the bolded words, because she will probably ask most of the questions based off of bolded words, right? And since you guys know how she makes exams, they're nothing too like deep, you know? She's pretty upfront with her questions. So keep that in mind. Don't like stress yourself out about understanding everything really well, because you just need to have a baseline understanding of these things. So yeah, I would just focus on the bolded words. Repetition is key here. I would try to look at the material like 3 times if you can, 3 or 4 times. Yeah. Yeah. Of course. I don't think so, no. Thanks, man.
If you find it, you can put it on there.
Yeah, cool, thanks. Yeah, so what's up?
It was just numbers 10 and 12. I was
Mhm.
wondering if you could go back.
Yeah, of course. I kind of screwed up the numbering on this one.
And where do we find this?
This is on ucfstudyunion.wordpress.com. Then you go to the biology section and you'll find my name.
Okay. Thanks for everything this
Yeah,
semester.
no, thank
I
you.
wanna see you're taking physics one, but we're in different sections, I think.
Yeah, shoot.
Yeah. I don't know.
I'll see you around.
I'll see you around.